So hello everyone. I hope you can hear me now. Can you hear me now? Yes, I guess. So yes, Julian said I'm going to be talking about SNP heritability, and in particular, what are we actually estimating when we estimate SNP heritability using kind of REML approaches. And so estimating heritability is kind of a thing quantitative genetics or statistical genetics has been doing for a very long time, pretty much from the very beginning of the field, starting with Golden, who kind of looked at the relationship in height between parents and children, and then going on to looking at comparison of other close relatives. At some point, somebody thought of the idea that you could use all types of relatives in one model using a pedigree and the kind of linear mixed model approach. And most recently, we've kind of reached the stage where we can genotype lots of individuals. And the idea has come up that rather than using a pedigree, you could estimate the relationships between individuals directly from the genotypes. And this kind of idea was introduced into human genetics, which is where I work. I know that in animal breeding, it kind of arose earlier with this paper by Young et al. In 2010, and it's been kind of super popular. So this paper has been cited like 4,000 times. The software has been cited another 4,500 times. And, you know, basically all papers estimate heritability when they do anything in humans. However, since this paper came out, there has been a kind of steady stream of other papers in, you know, kind of high impact journals like Nature Genetics and so on, which all have kind of said, well, this original model is kind of wrong. It doesn't give you quite the right thing. The estimates are not correct. And here's a better model, better method. Generally, by kind of constructing a slightly different genomic relationship matrix from the genotypes. And the kind of question that naturally arises, seeing that all these papers kind of claim that they do the right thing, but all are different, is which one is actually doing the right thing. All these papers have kind of formal arguments why they're doing the right thing. And basically, what I'm going to try to do in the rest of the talk is reconcile all these papers and actually explain why what they're doing and why I don't think that they're potentially doing the right thing, or at least not doing the right thing in the sense that you might expect. Before I get to that, though, many of you probably have been using kind of heritability estimation with kind of genomic relationship matrices computed from genotypes. And you might be kind of wondering what I'm talking about when I say the estimates are not quite right, or, you know, we need better estimates, because you probably have been doing it and all your estimates look really good and you're quite happy with it. So I'll give a very brief example of what I mean that these estimates are kind of behaving potentially funny and not in the way you might be expecting. And it's not an esoteric example. It's a kind of most straightforward example you could have in human genetics, estimating heritability of height. And I'm not going to do anything weird. I'm going to set up the analysis in the most standard way you could. So I'm just going to use common SNPs. I'm going to filter for Hardy-Weinberg equilibrium. I'm going to include the standard covariates. I'm going to remove related individuals, and I'm going to use a population which is quite homogeneous, which is kind of individuals of white British descent. And I've got about 300,000 of those individuals. And what I'll do is not just estimate one number, so one heritability for those 300,000 individuals, but instead I'll take different fixed sample sizes, and I'll split these 300,000 individuals in non-overlapping subsets of this size and get an estimate for each of these subsets. And what I'm plotting on the right is the kind of mean of these estimates and the variance of these estimates for different sample sizes. And as you can see, this kind of the expected value of this estimate changes. So the first thing you might think is, well, there's a bias. but it's maybe okay because asymptotically it will still estimate the right thing. Unfortunately, the kind of true heritability for this trait with these SNPs that are in the model is about here. So asymptotically, it's not really doing the right thing either. But still, you might not think this is particularly bothersome. So I'll do something else, which is maybe a bit weird, but I think shows a kind of interesting problem with these estimates. So what I'll do is I'll use the same data, but I'll, for each individual, copy the genotype several times. So each individual's genotype now consists of n copies of their original SNPs. What I do then is I take all the copies apart from the first one and permute them between individuals. So essentially an individual's genotype is now the original SNP genotype plus n copies from a random other individual. And obviously, these additional copies shouldn't contribute any heritability. They're just random genotypes which have no association by design with the phenotype of the individual. So what happens if I now start computing G-REMLs with these kind of extended genotypes? So if I start just with the original genotype, which I haven't permuted, obviously I get the same thing that I had in the previous graph, here replotted in blue and without the kind of variances. And then if I add one copy of permuted genotypes, I get this red line. And if I add 3 copies, so I've got 4 times the number of SNPs, I get this yellow line. So this is kind of great. If I feel that my heritability estimate is too low to publish, I just add random genotypes to my thing and I get a higher estimate. What's not to like? It makes it easier to publish. But obviously, this is probably not what you would have expected. So in the rest of the talk, I'll kind of explain why this is happening and why it's exactly what you should expect, and why none of the methods that have been published address this problem. Okay, so before I get into the details of that bit, I would like to talk about a kind of more general issue, which is, I think, very relevant here, because what I'm not saying is that these other papers are in any way wrong, especially the kind of math is, I'm very sure, correct. What I'm going to argue is that they kind of answer potentially the wrong question. So what is it that we're actually trying to do? I mean, I've gotten quite far so far without actually saying what I'm trying to estimate, or what my setup of what my overall goal of this whole process is. So what I will assume we want to do in statistical genetics, obviously other people might have other ideas what they want to do, but what I'll assume we want to do is we have some population in the real world out there, and this population is kind of governed by some process pi, and we're interested in kind of computing some property of the population to learn something about the population. So it could be something very simple, like we just want to know the expected height of an individual in the population, or in our case, we want to know what is the SNP heritability in that population out there. The problem obviously is that we've got little idea of what pi is. In particular, we can't just write down pi. It's basically somewhere out there in the real world. So we've got no way of directly computing X, the property that we're interested in. But what we can do is we can take a set of individuals from the population in the real world as a sample, and kind of propose some kind of model which has some parameters, and use that sample to fit that model to get a kind of theta hat, which are the kind of fitted parameters, and then use this fitted model to get X hat, which is an estimate of the property of interest. However, there's an important question which we kind of need to address is, what is the relationship between X hat and X? Okay, it is not immediately obvious. that if you pick just a model and you compute something from it, if you fit it to data from some other unknown process, that the quantity you get out is in any meaningful way related to the thing you were actually interested in. So you have to kind of look back, and that's what we'll try to do here, and relate your estimate back to the property that you're actually interested in of the real property of the real population in the real world. And I think that's what's kind of missing from the other papers, because what the other papers kind of do is they look at this setup where you have an assumed model and you ask, well, if I have this assumed model with some fixed parameters theta star, and I sample from this model, and I follow the same process of fitting, whatever my method does, how does the estimate in this relate to the thing I would get under my assumed model? This is a kind of related question. It's an interesting question, but what it doesn't tell you is anything beyond the bounds of your assumption. So you might get the result that, well, in this setup, my estimate is what I'm interested in. It's an unbiased estimate of what I'm interested in, but it doesn't tell you what would happen if I actually use the same method and apply it to a population which does not follow your assumed model. And in reality, none of the populations out there will follow any model that you assume. And to illustrate this kind of restriction of this approach, we can just look at one possible conclusion from it, which you can find in various papers out there, which is that if we have a linear mixed model with a genomic relationship matrix, then if the genomic relationship matrix was calculated from the causal SNPs, then we would get the correct estimate of heritability. This is right, but this is what would happen if we sampled from a linear mixed model with given parameters, and then fitted another linear mixed model with a slightly different G-REML, or the same G-REML, and got new estimates. However, if we want to look beyond data which comes from a linear mixed model, this result doesn't really apply. And we can show that by just simulating a population and a phenotype with known causal variance. And it's very easy to show that even if you use the G-REML, which is based on the causal variance, which you kind of recover the same behavior that we've seen for human height. So while the result itself is not wrong, it is just restricted to a very specific case and doesn't tell us anything or little about what would happen if we apply the method to a general population or general data from the real world. Obviously, the kind of more general question seems a bit daunting, because if we don't assume anything about the population, if we don't make any assumptions about this process pi, how can we even define what we're interested in? And the nice thing is we can for SNP heritability. So we can define the SNP heritability with respect to a general population governed by pi without making any assumptions about pi. So what is the SNP heritability? The SNP heritability is the variance of the additive genetic values or breeding values in animal breeding. What are these breeding values? They are just for an individual, the genotype times the SNP effects. What are the SNP effects? Well, the SNP effects are just the results of regression of genotypes on phenotype. And all you have there, you've got a variance and an expectation. Both are taken with respect to this unknown pi. But overall, we have written down a definition of the genetic variance or SNP genetic variance, that is the genetic variance captured by the set of SNPs in G. Obviously, we can't compute it because we don't know pi, but that's why we're here. What we want to do is see whether we can relate this quantity somehow to what we get from REML, which is below, which is this sigma hat, which is what we get if we take a sample of phenotypes Y and a sample of and associated genotypes, which we use to compute a genomic relationship matrix K, and fit this model by maximizing this kind of expression with respect to the two parameters sigma G and sigma E. And yeah, so in the rest of the talk, I'll basically show how we can relate these two quantities, sigma hat and sigma, directly to tell us what the estimate that we get from REML actually means in terms of real properties of a real population out there. I won't go through the details, so I'll just very quickly sketch where the result comes from in a kind of simple corner case, which is the asymptotic behavior. That is what happens if the sample size goes up and up and up towards infinity. And then I'll talk about the more general results and the kind of implications of it. So what I've written on here is the same equation as above in a slightly different form, which you might be familiar with. It's commonly the starting point for the linear mixed model or genetic linear mixed model, which is that you have a kind of regression, which is the first Gaussian under the integrals, which is kind of the phenotypes, the phenotypic mean plus the breeding values given by the multiplication of the genetic matrix G and kind of SNP effects with some Gaussian independent noise. And then you put a Gaussian prior on the SNP effects with some covariance matrix sigma. So what you normally do in REML is you integrate both the mean and the SNP effects out, and then you end up with the equation that we had before, with just one Gaussian and the genomic relationship matrix. But we'll kind of do something different. We'll not integrate these out, but we'll look at the expressions inside the integrals. And first we have a look at this first Gaussian without looking at the prior. And we can just rewrite this Gaussian. So we can essentially, in a Gaussian, the mean and the observation are in a sense symmetric. You can think of a Gaussian as either a distribution over the observation with a specific mean, or if you have observed a value, you can think of it as a distribution over the mean given the observation. And we do kind of the same thing here, just slightly more complicated because our mean is this kind of matrix vector product, and we just rewrite the Gaussian. So we haven't changed anything in the expression, but what we get is a new expression which is slightly more interpretable. In particular, if we look at it, we have a Gaussian over the SNP effects, which as the mean has the ordinary least squares solution to the SNP effects. And in the covariance, you see this R tilde, which just turns out to be the empirical covariance of the genotypes. So it's a kind of LD matrix between the SNPs. And the nice thing about this expression is we can look what happens to it when the sample size goes up. So because the beta tilde is just the ordinary least squares solution, we know that by standard results, it converges to the regression coefficients. And the second expression, because it's an unbiased estimator of the covariance, and it's divided by the sample size n, nicely converges to 0. So this first Gaussian, what happens to it when the sample size goes up? It's a Gaussian which is more and more centered on the true underlying regression coefficients in the population, and its variance goes down, so it concentrates more and more on this point. So what we'll do in a slightly hand-wavy fashion is that when the sample size is large enough, we'll just replace the Gaussian with an approximation which is just a point mass on the true regression coefficients, which is where it concentrates anyway. And if we do that, we notice that the resulting integral is very simple to solve because the first term is 0 everywhere apart from at the true regression coefficients, the true SNP effects. So the whole integral is just the evaluation of the second term at this point. And now we have this expression, which is just a Gaussian. It only depends on one parameter, the sigma g. The dependence on the sigma e has dropped out in the limit, which is nice. And if you look, if you write this expression out, the minimization is quite straightforward. It's just the mode of a chi-square distribution. And it is given by this. So it's essentially the betas times the inverse of the prior covariance of the SNP effects times the betas. So this is nice. This tells us that asymptotically, basically, our choice of sigma determines, in a sense, the expression of the estimate. Okay, so you can do this whole process kind of more formally and more carefully and without taking the limit. And you can get a kind of more general expression, and this is what you get. So you can write the REML estimate as essentially a function of several terms. You've got these b tildes there, which are just the ordinary least squares regression coefficients, which are unbiased estimates of the multiple regression effects of the SNPs. You've got this r tilde, which is the unbiased estimate of the LD matrix. You've got this m over n term, which is the ratio of the number of genetic variants you have in your model and the number of individuals. And you've got this lambda, which is kind of the term which means that this is not a closed form solution to sigma, to the REML problem, but it's kind of well behaved and it's just a bit of a nuisance, and it doesn't stop us to interpret this overall expression. So we can now go and look back at the expression we were actually interested in, so the population parameter sigma g, which we can very quickly rewrite into the following. The kind of SNP effects are, which is the covariance of the SNPs, so the LD matrix times the SNP effects. And as you see, these kind of two expressions are structurally very similar. In particular, on either side, you've got these betas and the beta tildes, and luckily the big beta tildes are unbiased estimates of the betas. So, that's kind of good. That matches what we kind of want. But you have this thing in the middle, where in the population, you should have, well, what you're interested in is the LD matrix of the population. But what you get in REML is this kind of more complicated matrix, which on the one hand does contain an estimate of this LD matrix, but on the other hand, it has this sigma in it, which is the prior covariance of the SNP effects that you chose, which is basically governed by the G-REML or the genetic relationship matrix that you choose. So if we look at this more closely, we see that these two terms are weighed, in particular by this m over n term. So in particular, when this term is large, what that means is that the overall expression kind of is dominated by your estimate of the LD matrix, which is good. This is what we're kind of hoping for, because this is the expression that appears in the kind of population quantity that we want. However, once this m over n term gets small, that is when the sample size grows and grows and grows, this overall term is dominated by our choice of prior for SNP effects, the sigma, which is what we saw in the asymptotic effects, where it basically reduces completely to just the sigma. So in summary, what we can kind of conclude is that if we're interested in a property of a real population and we just fit a REML model to a sample from that population, the quantity that we're estimating in some sense changes with the ratio of the number of variants and number of samples we have. In particular, and this is maybe a bit counterintuitive because it goes against the kind of normal assumption that, in the limit of large samples, priors don't play a role anymore. What we have here is that actually, as the sample size grows and grows and grows, the prior that you chose for the SNP effects becomes more and more important and dominates, in a sense, the quantity that you're estimating. Importantly, if you're interested in actually estimating the population property, what determines whether you are estimating it or not is whether this sigma, the prior on the SNP effects, models the LD structure well, not the distribution of effects, which is good because while the latter, so the assumption that you need to model the distribution of effects is what is kind of generally assumed in the literature, this is a trait-specific parameter which would change from trait to trait. Different traits have different distributions of SNP effects. But the LD is the population parameter, which is constant across traits. So we have a kind of hope that we can pick a G-REML, which we can use which would give good results for different traits. We wouldn't have to change the G-REML from trait to trait. And we can look at two alternative G-REML choices. On the one hand, the standard G-REML, which was first proposed, I believe, by Van Raden, and which corresponds to a choice of an identity matrix for your sigma. And if we basically plug it in and look what happens, asymptotically, you are essentially estimating the what's called the genic variance, which is just the sum of squares of the SNP effects. Alternatively, we can pick our sigma to be the inverse of the kind of an estimate of the LD structure, and I'll term that LD structured G-REML, and it's something that's been actually proposed in 2018 by Matthew et al. Without a formal argument. And if we do that, what we see is that asymptotically, we would actually estimate the genetic variance of the population. So when would that matter? It would matter if there's a LD contribution to the genetic variance. So if we look at the genetic variance, written here again, you can break it up into two parts. One is essentially the diagonal, in a sense, which is just the sum of squares of the SNP effects. And then you've got a second, and this is always positive, this is called the genic variance. And then you've got the second term, which are basically the cross effects between different SNPs. And it depends on the LD between these SNPs. And this term can be either positive or negative, depending on how the LD and the individual SNP effects line up. And the question is that something that we would expect to arise in natural conditions? Because if we expect the contribution to always be zero, then obviously, this doesn't matter because our standard G-REML kind of only estimates the first part, so everything should be good. There's a very simple case where you would expect a positive contribution from LD, and that's assortative mating. So if you have a population where for a certain phenotype, the mates are more likely to be, or are more similar than they would be expected under kind of random assortment, then what happens is over the generations, you're building up additional genetic variance, and this additional genetic variance doesn't come from changes in the allele frequencies of the causal SNP, but it comes from buildup of a certain type of LD between the causal variants. So what we can do to kind of confirm our theoretical ponderings is we can just simulate populations under assortative mating, which is what I did, with different kind of genetic architectures for the trait under assortment. So from left to right, you have different levels of polygenicity. So either 0.1% of all SNPs are causal, or all SNPs are causal, meaning like a fully polygenic trait. And at the top, you have traits where the causal variants are rare, that is, their minor allele frequency is under 5%. And at the bottom, you've got more common causal SNPs. So all causal SNPs have minor allele frequency more than 5%. And I estimate heritabilities with different sample sizes again, using two types of GRMs, the two types discussed, the kind of standard GRM that Van Raden proposed, and this LD structured one. And it's kind of exactly what theory predicts. So first, for the LD structured one, you get kind of estimates throughout the range of sample sizes, which are pretty much close to what your simulated heritabilities are. Those are the red lines. And the blue lines, which is the standard GRMs, you see that you have the same effect that we saw in human height data, where for smaller sample sizes, you kind of overestimate the heritability, and then your estimate drops towards the genic variance as the sample size increases. This is exactly what you would expect if you look more closely at the kind of expression and think through what would happen if you had a positive contribution from LD. Okay, so that's obviously nice that we kind of can explain what happens under assortative mating, but you might be thinking, well, assortative mating is a kind of outlier, you know, most traits are not under assortative mating. So I generally assume that I don't have any contribution from LD. But you have to be kind of very careful there, because the kind of result has some subtleties. So one of them is, for example, that all the expectations we took were under the sampling distribution. So our pi is, in a way, the distribution from which we get our observation. So it includes any kind of additional filters or anything like that that we put on top of the natural processes governing the population. So for example, if we do case control sampling, even though in our underlying population, the kind of causal SNPs might be in perfect linkage equilibrium, and there might be no contribution of LD. Once we kind of put that natural population through our case control sampling filter, you're going to see Bolmer's effect, which kind of induces linkage disequilibrium in that kind of case control population between the causal variants. Another one is that you might be thinking, well, my kind of causal SNPs are very far apart, so they will be in rough linkage equilibrium. There's really no reason to think that there should be any LD between them, so I can just ignore this contribution from LD. But the important thing is that these betas in our thing, they're not the causal effects, they're the regression coefficients on the SNPs that we included in the model. So even if your causal SNPs are very far apart, if they're not actually included in the model, what you're dealing with is SNP effects due to tagging of causal SNPs. And the question that kind of arises is, what happens there? Can that lead to a contribution from LD? Even so, because if it did, that's kind of important, because this is pretty much the standard setup. You don't have your causal SNPs in the model because you don't know what they are. And you're basically capturing tagging. So what do I mean by tagging? Very quickly, if you have a QTL and it has an effect and the surrounding SNPs have basically no causal effect, if you're not observing that QTL, so it's not in your data, your betas will be basically this effect of the causal QTL redistributed through LD onto the surrounding SNPs. So now you have a set of SNPs which are in LD and which all have a non-zero effect. So if these things are kind of in any way coordinated, you'll either have a positive or negative contribution from LD. So what we can do is we can look at what happens under different assumptions about how QTLs behave. And it's been kind of argued that QTLs are not randomly distributed with respect to how well they're tagged by the surrounding SNPs due to processes like selection and so on. And so what I did is I took I simulated traits based on QTLs, which were sampled from different kind of tagging bins. So you either took just QTLs, which were SNPs, which were very weakly tagged by the surrounding SNPs, or very strongly. That means in very high LD with the surrounding SNPs. And what's here is the kind of underlying variance generated by the causal SNPs. If you look at the kind of genetic variance captured by the SNPs in the model panel, which do not include the causal SNPs, you see what you would kind of expect. If you have very weakly tagged causal SNPs, obviously you can't estimate the entire genetic variance, you only estimate a smaller portion of it, which is tagged by the surrounding SNPs. Whereas if you've got very strongly tagged causal SNPs, you will pretty much recover the entire genetic variance. But what's interesting is if you break this up into the two components of genetic variance and LD contributions, the architecture that changes dramatically between the kind of very weak tagging scenario and the very strong tagging scenario. So you can see that for very weak tagging, you have a genetic variance which actually overestimates the captured genetic variance, and you have a negative LD contribution. Whereas if you look at the very strongly tagged QTLs scenario, then essentially the genetic variance captures the entire genetic variance, and there's very little contribution due to LD. And if we do the same thing of estimating for different sample sizes, we see exactly what we would predict. So if you look at the very weak scenario with the negative contribution from LD, you see this kind of increasing estimates of heritability with sample size. Conversely, if you look at the other end with the very strongly tagged ones, you see the same that we saw in the assortative mating scenario or human height, where you've got these decreasing estimates as sample size increases. And if you use the LD structured G-REML, you get basically constant estimates over sample sizes as the theory would predict. So to come back to the kind of original example, this behavior of decreasing estimates, which kind of undershoot the true heritability, should not really be surprising. Height is known to be under assortative mating in humans. So this is pretty much exactly what we would have expected for a trait under assortative mating. And we can now kind of conclude that the asymptotic heritability is just going to be the genic variance of height for the SNPs in the model. The same with this kind of slightly weird thing where I added the permuted genotypes. Obviously, what's going on here is I've shifted us in this space of ratio of number of individuals to number of markers. So while the underlying heritability doesn't change, I'm kind of moving in this space of what quantity I'm estimating. And I can very simply visualize that by plotting the same data, not as a function of sample size, but as a function of the ratio of sample size to number of markers in the model. And you can see that essentially all three models estimate the same thing for the same ratio. So if we very briefly look back at the other papers and what they have basically proposed as solutions and why it potentially doesn't work. So on the one hand, you've got the idea of stratifying SNPs into multiple G-REMLs by linkage, disequilibrium, and math, which is this G-REML LDMS idea. And you can actually show, if you go through the mass for models with multiple variance components, that asymptotically, actually, this thing will result in the same estimate that you would have with just one G-REML. So while the estimates might look different, as the sample size increases, they will actually converge to the same thing. Similarly, if we look at LDAC, which is another proposal of in human literature, which is based on essentially using a G-REML, which puts a different prior on the SNP effects, kind of arguing that what we need to do is match the true distribution of SNP effects with our prior to get the correct estimate. If we apply this to traits with positive LD contribution or negative LD contribution, we see that it kind of has some erratic behavior consistent with the theory of what would happen if you had a diagonal sigma with some weights in it. So that's essentially it. I'll just briefly run through a set of questions that usually come up at the end. And I'll briefly address those, and then we can maybe get to more general questions if anybody has any. So what quite often comes up when I talk about this is that somebody basically says, well, this is in direct contradiction to some result about G-REML. And as I say, I don't think any of those papers are wrong. I think they just answer a different question. Usually when you then look at the paper the person refers to, is those papers generally start with the assumption, assume that my population is sampled from this linear mixed model, and then I apply REML, what do I get? This is not the question we address here. The other thing is, you might say, well, this is all great, but my breeding value predictions are actually quite good with REML, so I have never seen a problem. I haven't spoken about breeding value estimation or prediction. That's a completely separate topic. This is very specific about estimating the parameter SNP heritability. Another thing which people kind of complain about is that I only talk about asymptotic results, or things which should only work when sample size is larger than the number of markers. Actually, in the paper, we have results which span the whole spectrum. So you can extend this result, which, if you look at it closely in the slides, depends on inverting a matrix, which is only invertible when n is bigger than m. You can extend this result to the full spectrum of sample sizes and marker numbers. It just gets kind of more tricky to interpret and explain. So you can go to the paper and it's all there. Other people basically say this is just bias variance trade-off for any model with regularization, and there's no way around it if n is smaller than m, and so just live with it. The thing here is, I'm not saying that you want an unbiased model. I think bias is sometimes good, especially when you trade it off against variance. The problem to trade bias against variance is that you first need to know what the bias and the variance is to know what trade-off you're actually making. I also don't say anything about methods which are not REML. This is very specific. This is an analysis of REML. So I don't really have anything to say about other methods, but I think it would be useful if somebody looked at the other methods and went through similar analysis. And in fact, if anybody is interested in looking at other methods together, get in touch and we can have a look at them. I do not propose a practical solution, indeed. That's not the aim here. It's just an analysis of an existing system. Yeah. And then I guess the most important one is people ask whether I'm suggesting that all previous REML estimates or results rather are wrong. Yes and no. I think they are wrong, but I don't think it matters. The reason is that if you have small sample sizes, your errors will be so large that, to be honest, it doesn't really matter where your central estimate sits anyway. You're pretty much covering most of the spectrum of heritability anyway. If you've got very large sample sizes, whether your estimate is, slightly off in one direction or the other, if your argument really depends on being off a few percentage points in one direction or the other, then you've probably got bigger problems with your argument than the fact that your estimate is slightly biased. So yes, that's pretty much all I wanted to say. And thank you very much for listening. This has been, to some extent, joint work with John Williams, who asked the original question about whether we could work out what REML actually estimates. And I'm happy to take any questions. Thank you.
Thank you, Konrad, for your nice talk. In case anyone has any questions and Konrad hasn't answered them yet, just drop a message in the chat or unmute yourself. So far we have nothing, so okay. Again, Konrad and me are in the same room if anyone is surprised why I look over my computer all the time. So one question I would have, how easy is it to implement, I continue with my question. And how easy is it to implement these LD corrected genomic relationship matrices?
Okay, so as I said in my questions, I do not propose a practical solutions. The LD structured G-REMLs are kind of nice in theory. In practice, to estimate the LD matrix, you need more, because you need to invert it. You need more individuals than you have markers, which is not the setup we are currently in, unless we increase our sample sizes rather massively. So they're in no way practical. They're a kind of theoretical result, but they are not a practical solution to these issues.
Okay, I'll mute myself again. We have a question from Gregor. So Gregor says, great work. I truly hope it gets picked up in the literature. In the effective tagging, you showed a partition into genic variants and LD covariants. Do you have estimating equations for each of these components? We have addressed these by using samples from posterior distribution of betas, but it can be costly.
Yes, so do I have expressions for these? Do I have estimating equations for these? Or that was the question. I kind of do, and Gregor knows it because he's emailed me about it before. I think the correct way is to do what you're doing, Gregor, of getting the estimates directly via the estimates of the SNP effects, or rather the estimates of the breeding values. And it's just a question whether you sample, which is what you're doing, or you compute them in closed form. As I say, you can compute them in closed form if you assume the kind of linear mixed model. And I will get those equations to you when I have a bit of time.
And there's a follow-up question from Gregor again. Konrad, can you clarify why this is not a practical solution? Which bit is missing? Gregor, if you want, you can also unmute yourself and just ask ahead. Or
okay, why is it not a practical solution? So let me go. So to compute this LD structured G-REML, what you need is, well, there's two ways of computing it. Because essentially what it's doing is another way of thinking about it is, rather than using this prior, which is the inverse of the LD structure, is to you're fitting a model where you're using orthogonalized genotypes. So you take your genotype matrix, and rather than standardizing the genotypes, which is what you commonly would do, is you fully orthogonalize them. So you essentially take the singular value decomposition and just use the first term of that, right? The problem with that, obviously, is that if that works fine if you have more individuals than markers, because your genotype matrix is not full rank, and everything is good. If you, in the other setting where you've got fewer individuals than genetic markers, then you're going to end up with the G-REML, which is basically the identity matrix, and it makes the model non-identifiable, because obviously the G-REML and your residual matrix are the same. An alternative way of thinking about it is you need to compute the inverse of the covariance of SNPs, i.e. the inverse of the LD matrix. Obviously, if you have fewer individuals than markers, then the covariance, which is or the covariance of genotype, of the covariance between the markers, which is of size number of markers by number of markers, will be not full rank, and you can't invert it. It will be singular. So that's why it's not practical.
Okay.
Thanks.
Then 2 more questions from Ivan. So he says, great talk. First of all, what are the computational properties? How do you implement this to solve for 1 million samples or individuals?
Well, as I'll return to my questions, which commonly get asked. Oh, sorry. Where are we? I'm not proposing a practical solution. This is not a new method to compute heritability estimates. It's an explanation of what happens in other methods which have already been proposed. So I have no proposal in how to compute heritability estimates with more individuals or more markers. But I can tell you that if you compute your heritability estimates using like GCTA or AS-REML or whatever with a particular genomic relationship matrix, what those estimates mean and how they relate to real properties of the real population that you're probably interested in is in the real world.
So then his follow-up question is, in animals we found genomic estimates be biased when we have selective genotyping. This was partially improved by using combined reduced matrix H single-step approach. Have you tried to compare estimates with this kind of setup?
Well, I don't actually need to compare estimates. So I don't know off the top of my head what specific form of genomic or of relationship matrix in general you're referring. If you're interested, get in touch with me and we can have a look at it.
Okay, thank you, Konrad. Yeah, Ivan says thanks as well. So I think there are no more questions. Thank you very much for your talk. To anyone else, thank you very much for attending today, and we will have the next talk in 3 weeks, which topic to be announced. So have a nice day and see you hopefully in 3 weeks. Bye.